Francis Crick was a British molecular biologist, biophysicist, and neuroscientist. He formulated the Adaptor Hypothesis in the 1950s. Crick used this hypothesis to propose the function of tRNA in the cell. He chose this name because he believed that there would not be a sufficient affinity between amino acids and nucleic acids for protein synthesis. He found that with an adaptor molecule, specific hydrogen bonding could occur between nucleic acids and specific covalent bonding could occur between an amino acid and nucleic acid tRNA.
First, take a look at our central dogma. Proteins are synthesized by RNA by translation, and RNA is synthesized by DNA through transcription. DNA and RNA are compatible systems since they are both made out of nucleotides, but RNA and proteins are not compatible, so the use of ribosomes and our adaptor molecules must be used for translation to occur. Let's look into translation. Let's look at an analogy to help us better understand adaptor hypothesis. Imagine that there are many confused passengers about to board a plane because they do not have a ticket. They ask the flight attendant for help to find which seat is theirs. In this case, the flight attendant, which symbolizes tRNA or adaptor, helps guide each passenger, which symbolizes the amino acid, to the correct seat, which symbolizes the ribosome. Crick had this idea about the tRNA molecule. tRNA, the green, which is the adapter, acts as the attendant in the situation to guide the amino acid, the passenger, and according to the mRNA, and bring it to the ribosome, the seat on the plane, which fits like a puzzle piece. In the ribosome, it enters the A site. Then it links to the amino acid in the P site and exits out the E site. In summary, the tRNA acts as an adapter connecting two things directly between the mRNA and the amino acids. The anticodon in blue on the tRNA determines the appropriate amino acid. The anticodon base pairs with the mRNA sequence codon in purple and deposits the amino acid into the polypeptide chain. Let's look at the universal genetic code. Perhaps an alteration occurs that prevents tRNA from acting as an adapter. In this case, this means that there could be an improper crossover or there was a damage to the DNA that was caused from an outside factor. This can cause the resulting mRNA to change, producing the wrong amino acid. For our first example, we can look at isoleucine, shown in purple, which can be coded as AUU or AUC. If the alteration happens in the first two letters of the code, it will code a different amino acid. Other alterations can occur from the start or the stop codon. If the start codon is altered, the consequences is that the entire mRNA strand can go uncoded and the protein cannot be made properly. If the stop codon is altered, meaning that it possibly was mistaken to be an amino acid, it can result in a longer protein to be made.